Another method that is now one of the gold standards for trajectory analysis is the update of the MST, which is a graph abstraction, or the principal tree learning. So this is one of like DDR3, and others are inside this category. And most current methods use this method. So the problem with MST is that if you have all those data points connected, so for example, like these lines here all connect this point and is the best way to connect these data set, for example. But if you have a shift in one of the cells like this, then the trajectory completely changes, because now it's another completely different tree is the best way to connect all those cells. So to avoid these problems, or like depending on a single data point, now what we can do is to find local clusters in your data, like here and here, and then find a minimum spanning tree among the clusters. So then the clusters will always be connected like this, because the clusters now are more robust to these small changes. So even if I change this cell up here, this is still going to be one cluster, this is still going to be another cluster, and the connection between them will always remain the same. And basically, that's the concept of graph abstraction. So defining these clusters and then creating a connection between them based on proximity. So like these clusters are close to each other. Of course, they are not connecting to something far apart. So this is the idea of the DDR3 or any other graph abstraction method. So here in this example is just the DDR3 that you see in Monocle version 2. You have your raw data, and then you do a dimensionality reduction via diffusion maps, ICA or TSNE or UMAP. And then you try to fit these cluster centroids in your data and connect all those clusters. And then you assign the cells to these trajectory lines where you define your trajectory, and then they are here. And then what you can do is update this trajectory back to your initial dimension and compare. Are the cells that I fit in my trajectory close to my real data? If not, then I need to update a little bit the trajectory so that the trajectory fits better my data. So this is an interactive process that happens in Monocle 2, but it's very similar to what we see in other methods as well. So after you find the best representation of your data, so for example, here in the first representation, you see that there's this line sticking out. So maybe actually you don't need that line for your data, to represent your data. You just need this bifurcation here. And one thing that is very important now to say as well is that in trajectory analysis, you can never find what is the start or the end. You can predict or try to do that, but you always need biology in that moment to find where should I start my trajectory. So here you need to select a route. It could be here, it could be here, but that you need to look for the marker genes for your cell type. So markers of undifferentiation, or markers for the progenitor cells, and so on. And then the pseudo-time trajectory is basically the distance in this line from the selecting point that you have. Like I said in the beginning, so it's an abstraction of time. So it basically here in the graph abstraction, it means the distance from the selected point of origin. And most methods will have ways of finding these branching trees for you. So you don't have to really think about that much. So all methods that came after Monocle 2 are based in this kind of same idea. So you'll see in the exercises, we're giving an introduction to PAGA and Slingshot, which are quite flexible methods. PAGA is based on a graph abstraction method. So you create a graph out of your data and you project it and this is a very similar approach as UMAP, for example. And then from that, you connect the clusters that are present in your data as the graph abstraction that I just mentioned about. And from that, you can define where is your progenitor based on some markers, which are here. They're labeled stem. And then from that, you can define the trajectory for here, and through all these paths in your graph. And for slingshot, is exactly the same concept. So if you have your data, then you do define some clusters throughout the whole data space, as long as they are connected like this. And then from the cluster centroids, you can connect all the different cell lineages that you have there. I just can also mention that these techniques are also done in most of the other current methods for trajectory analysis. So, I want to test T-scan. T-scan uses PCA space and also this cluster representation analysis. Cell router uses TSNE, and then you just basically change which dimensionality reduction you use or which clustering method are you using, but overall they are very similar in composition. And just another example, you probably already heard of Monocle 3, maybe, for trajectory analysis, and it's exactly the same concept. So you do normalization and PCA, do clustering using Louvain, dimensionality reduction using UMAP, and then you construct your clusters, partition your cluster, or like your cluster centroids, like I said in the previous slide, connecting which cluster is closer to which cluster. And then you find the tree using this DDR tree or the learn the graph. And then you find your pseudo-time assignment, like here. So you define what is your root, you have your connected graph. So then the pseudo-time will be the distance from the root. And then you can do differential analysis or differential expression.